Okay, so this is a continuation of the chapter 4, cells and organelles. So we're talking about this topic. How did the first cells come to be? And I'm going to summarize what we did in 1 minute and then go to the next step to finish this part, this first point. So in the first section, we talk about this. Yeah, how inheritance material appear. And we start even earlier than that. Yeah, that it was how simple inorganic molecules start getting together to form macromolecules that they were organic molecules. Now the next component that is fundamental for life to appear is to create an inner environment that separate the exterior to the interior, yeah, and where these macromolecules will start having a metabolism, yeah, and having their own environment that they can grow and have their own reactions, and all of that can be done with this. That is a simple vesicle with lipids that it create an interior here and an exterior outside. Okay, so interior and exterior. And during the semester, we will talk about how, if you see these different structure, the different cells, eukaryotic cells are formed by organelles, and many of those organelles will have this type of membranes and as a result of that, I wanted, as I said before, that you think about a cell like a very dynamic structure, like this city that things are moving in and out and in between them. Yeah, the same thing happened in the cell, that you have always a traffic of vesicles that are, again, these lipid structures. So, so how this happened? This is the question of this. We're going to put all these hypotheses together during this class. Okay, so we are going to start with this question. What is a liposome? That is pretty easy, and if you read the textbook, it will be even easier. If not, even if you know the Greek roots, it will help you a lot. So let's split it into two. So one section is lipo, and the other one is some. So some here means body, yeah. So they are talking about a structure, or yeah, let's put body here, yeah, and here lipo means lipid, so is a structure formed by lipids. Good, so A is incorrect. Small cellular structure found in the nucleus, uh, not only in the nucleus, and it is more found in the cytoplasm, so this is wrong. The idea that RNA was first, no. They believe that RNA, no, and spherical vesicles composed of lipid bilayers. Okay, so in this lecture we're going to talk about liposomes. Later we will talk about variation of those structures. That now this is a hot topic in biology, and we will talk about why. Okay, and why we're talking about lipids. So, as I said before, we talk about all these different sections, how from inorganic compounds we form organic compounds to later form polymers. And now, so we need to enclose all those structures, and to do that, we need these lipids that they are going to um, uh, self-organize and close themselves into this structure that they allow, again, to create an internal environment and an external environment, that it will be very important to create a metabolism, and here at the end to be able to develop self-replication. Okay, so let's talk about a little bit more about liposomes. So here you can see the lipid bilayer, that it will be located here. This is a photograph of a real liposome, and here inside, in this inside of the lipid bilayer, you can find here the liposome, and in the liposome you can have different molecules. So for example, it's very common to find, for example, RNA. And for that reason, we think that the primitive cells could have used that tool. And you can think that, okay, this is an interesting fact, but it's not important right now for science. However, fortunately, this is actually a hot topic in research, and it's very important. Not only it was not important a few years ago, but it will be very important in the future, and I will show you why. So you can look for this article. So it's from March 2021. I don't know when are you watching this video. Maybe it's already getting old. But the idea is, if you will read it with me, without these lipid shells, yeah, they would have not been the messenger RNA vaccines for COVID-19. Yeah. So if you put RNA to somebody, yeah, possibly is going to be degraded, is not going to go to the right place. Yeah. You need to have this lipid shelf in the same way that in the first cells you needed to protect them so they don't get degraded, that they go to the right place. You need to have lipid shells so they go inside of the cell and they are able to be uh, transcribed and the vaccine work. Okay, so I'm going to show you a small video, and later during the class, during the semester, we will talk more about this because this will become or is becoming a hot topic in research, and we are going to, instead of having vaccines, messenger RNA vaccines for COVID, we are going to do the same thing for cancer. So we know that very important target genes that we need to repair, and treatments could become much easier if we repair those genes. So if we if we put in a liposome here, we add the correct copy of the gene. Uh, Maybe this could help to heal the worst effect of cancer, and we are already studying those kind of things. Okay, so this liposome, yeah, these lipid vesicles are very important, and now I'm not going to tell you more. I'm going to show you the video.
In the late 1970s, scientists discovered an interesting property of phospholipids. That is, when they are hydrated, the hydrophobic tails mutually attract, while the hydrophilic heads seek the aqueous medium, thus forming a phospholipid bilayer around an aqueous core. The lipid bilayers then seal off to form a liposome. Since liposomes are composed of natural lipids which are biodegradable and biologically inert, their toxicity is minimal. The amphotericin B molecule is a good candidate for liposome-based drug delivery because it has a useful duality. On one side, it has a polyhydroxyl chain which is hydrophilic, and on the other side, it has a polyene hydrocarbon chain which is lipophilic, which allows the amphotericin B molecule to be encapsulated between the double lipid layers of a liposome. However, creating a true liposomal preparation is expensive and involves several complex processes. Cipla is only one of the few companies in the world which have successfully developed this advanced liposomal technology.
Okay, so that was not a promotional video. I'm for that you buy things from that company. What I just wanted to show you that this is not a theoretical concept. There are already companies working on that and trying to sell products on that. Rather than say that this is a good or bad company, I thought that it was very clear what they were explaining. That in the present, yeah, we can explore this potential that have these vesicles to transport different molecules inside, and there they show you how they can be located, for example, in the membrane. They can also be inside, and they are biodegradable. They are not, yeah, so they had a lot of applications, and later we will talk more about that. Currently, or for this class, I wanted that we think about the importance of the past. So, for the origin of life, so pretty easy. Imagine life start in the ocean, and if you have a big tide, if it was raining, you might have some reaction, chemical reaction. But if a tide comes, yeah, it will break. It will all these chemicals will separate. They will be far from each other. It will be very difficult that this early metabolism appear. However, when you have them inside of this spheric shelf, then the metabolism can start getting more regular and the first protocells start appearing. Okay, so these liposomes that we will talk more during the semester, they are important not only for the questions of the first primitive protocells, but they are also important currently and for the future to put some specific molecules or some specific RNAs inside of a cell. Okay, that this will be key for curing many diseases. Okay, let's summarize. I just told you 3 points. First point is that currently, yeah, that maybe you might be working in a company that it will be using lipids, yeah, that have produced these hollow membrane-bound vesicles that are liposomes. And although they were important right now for many scientific research, in the past, yeah, they could have also been important. Why? Because those liposomes, under some circumstances, they're able to carry out simple metabolic reactions. So, if the molecules inside here are able to have to react to each other, they will react there and they can create the simple metabolism. And finally, the scientists consider that these primordial lipids, they have been very important in early oceans because they were trapping the RNAs and they were forming the first protocells. Okay, so again, so we went through different steps of how life started, and what I show you is that the steps, they were not impossible. Actually, they were very likely to happen. If you put a lipid, oil in water, what you notice is that they form these these clumps and they group, yeah, and you don't require to have energy to do that. They do it just because it's the most stable stage. Yeah, so same thing happened with these liposomes that, as we watch in the video, they just self-organize into that. Okay, so with this we finish the section. I'm going to show you the answer of the question of what is a liposome. So now I'm sure that you know that a liposome, if you divide it into two, you know that this comes from lipid, the first section, and the sum means body, and all of these questions related to the DNA or RNA, all of these ones are wrong, and they are spherical vesicles composed of lipid bilayers, and we will talk about a structure similar to this ones in future lectures. Was just an introduction. And with this, we finish, if you remember, what came first, the chicken of the egg, this common sentence that we hear always, that we can translate it or think about that, but in terms of inheritance, yeah, and protein functions, and that in the origin of life is not a simple answer. And we try to show you different lines of evidence and try to think that not only even the DNA, the proteins were important, but also the RNA, and also the presence of membranes are very important. And we will talk more about that during the semester. And again, if you're interested in this topic, I will show you sections of this lecture in future section of the semester. They're very interesting, and if you like this topic, he is one of the person that knows the most about the origin of cellular life on our planet. So here is the link in case that you want to watch it. Okay, so we finished the longest part of this chapter. Why? Because I think this was new for you, a lot of these things. Uh, and here, we're going to move to the properties and strategies of cells. And here, there will be a lot of repetition, and I'm going to show you some slides that I'm going to say, if you know this, you need to review it. I'm not going to cover it in detail. Why? Because you have studied this since possibly elementary school. So if you have any doubt or you need to remember something, the textbook can help you. So for example, the slides that you see a little bit yellow is that I am not going to talk a lot about that because I know that cells have characteristics like complexity, molecular components, size, shapes, and specialization. Yeah, and we already talked about a little bit in the previous section. So I just wanted to show you that in this kind of slides, I'm just going to talk about that, leave it for you, and you can review it in more detail if you have any questions. So for example, I'm not going to tell you what is the main difference between prokaryotes and eukaryotes. I'm sure that you're tired that I tell you what is that, but the slide is there in case that you need to remember. Your textbook is there. So I'm sure that you remember for when you took your introductory biology courses that there were three domains of life: bacteria, archaea, and eukarya. And you can review that. I'm sure that you remember that. What I'm going to talk in more detail about these groups is how close are one to each other. And in this class, I'm going to show you the cellular basis of some of those differences and some similarities. Okay, so uh, we already talked about how the first cell could have appeared. I didn't go into a lot of detail of the properties and strategies of cells. I'm sure that you already know that information, and if not, you can find it in the textbook. Now I'm going to go into more detail, as I said, about this, the difference between these three domains. So the key point here is that I want that you observe these two phylogenetic trees, yeah? And here, this information is relatively new in the sense that when I was in the university, they taught it in a different way. Yeah, so first phylogenetic tree, I want that you see how close is bacteria to archaea and how close is archaea to eukarya. Okay, so if you see it here, you see that archaea and eukarya, in some degree, they are closer than bacteria to archaea. Okay, so although bacteria are unicellular and are prokaryotes, so there is some resemblance to archaea, they are not as close as we thought before. Yeah, and if you see the second one, the second phylogenetic tree, you have bacteria here, eukarya here, and archaea here. And this phylogenetic tree is showing you that they are separated. Yeah, that our bacteria and archaea are different, and they are as different as bacteria and eukarya from archaea. Okay, so let me summarize those points. And the idea is that so it seems that these three domains are very different from each other. Yeah, and in the following slides, I'm going to show you the cellular basis of those changes. So first thing that I told you is that eukarya and bacteria diverged long time ago. Yeah, and that is clear here. Bacteria here, eukarya here, and the divergence was here, while the divergence with eukarya seems to be here. In other words, although archaea resemble bacteria because they are both unicellular and they both are prokaryotes, archaea and eukarya they seem to be closer together. In other words, these to be prokaryotes, it doesn't mean that they are related to each other. Based on this phylogenetic tree, also the new information that is showing us is that actually bacteria and archaea are as divergent from one each another as human and bacteria. Okay, so they are much farther than we expect, and this is why we recognize that there are three different domains. So we were talking about this idea that is key, that there is a changing view of prokaryotes. In other words, we talk about that the term prokaryotes is not good enough. Yeah, that two organisms don't have nucleus. It doesn't mean that in the phylogeny they are close to each other. Yeah, this is just a gross structural feature, and is not necessarily an evidence that they are close to each other. Yeah, that they are related. So all the bacteria they will be close together, and all the archaea they will be close together. And it doesn't mean that they will be bacteria and archaea. They will be related. So when I was learning this topic in university, they actually they could have even sometimes a Venn diagram that you say there was an intersection because they were very close to each other. What different analysis have shown is that they are more independent than we thought. So, for example, if you analyze a way to analyze similarities and difference in phylogenies is using ribosomal RNA. So you look the sequence, and this sequence are a perfect way. They don't change a lot, so they are a perfect way to analyze how close or how far are different organisms. And they found that the prokaryotic cells are can be divided into groups that are very different. One that is bacteria, and another one that is archaea. So again, the changing view of prokaryotes is that the term prokaryote it doesn't mean that those two groups, those two domains, are closely related. Okay, so let's read this question together. This is a question that is at the level of elementary or high school, and then I will ask another question that is at the universal level. So be careful with that. So, what is a distinguished feature of this eukaryotes? So here we have one group that are the eukaryotes, group number one, and a distinguished compare to bacteria and archaea. Okay, so these two are two groups. So we have been talking that in elementary and high school they told you eukarya and bacteria and archaea are different, yeah. So here, based on the information that you had from high school, you will say that is the presence of the nucleus, yeah. It's not the lack of cell walls, it's not a unique membrane lipids. Maybe you don't know if they what are the membrane lipids of archaea, yeah, or bacteria, what are those? The use of chlorophyll for photosynthesis. So you know those maybe are not the answer, yeah. So at the level of uh, elementary school and high school, this is pretty easy. Okay, so then this you divide these two groups, the eukaryotes and the prokaryotes. The presence of a true nucleus is the difference, but at the cellular level of a at the level of a university, I will show you that as I have been talking. You go in more detail and you can see that there are unexpected difference. So I'm going to show you a table from your textbook, and I will show you in the beginning similarities, and then that when you study those in more detail, those organelles in more detail, there is more different that you expect. So again, I'm sure that you know the answer of the question, and you didn't need it. even to come to class. However, for the test in your textbook, they can show you that that question could have been more complicated. Here, for example, uh, in this table, they summarize all the different properties of bacteria, archaea, and eukaryotic cells. Good. So here, what I'm going to show you is very general features in the beginning, and you will see that in all of them, bacteria and archaea will be similar. Yeah, but very general. The idea that to show you those adaptations to be small. Yeah, that is what happens. So it's not that they are closely related, it's just that they are small. So this is what happens when you have small cells. So let me show you an example. So I already showed you that when you compare uh, the size of the bacteria and archaea are small, while the eukaryotes are much bigger. So when you compare the nucleus, as we said before, they don't have it, while the eukarya are similar. So this is what we thought about that. When you compare the presence of proteins that are important for the structure and movement of cells. So here we are a little bit biased. So we are going to tell you that they don't have actin and proteins, actin and tubulin. Yeah, they don't have those protein for movement and cellular structure. They have something similar. Yeah, but they don't tell us which one they have. And later we will show you that they have different ones. So the three groups have different variation of that. So, but here, from a very general perspective, this one don't have it, and this one have it. Again, endocytosis and exocytosis, this one eukaryotes have it, this one don't have it. But again, if you are a small cell, it's very different when that you are a big cell and you need to exchange a lot of materials. And again, these are only similar features. Now I'm going to don't talk about cell wall. The next one, I'm going to talk about the mode of division. Again, small cells have binary fission, while the eukaryotes, what they have, the thing that you have studied in possibly every course of biology, mitosis and meiosis, and you're tired of that. And finally, the typical form of chromosomal DNA is that here they are usually circular, while this one is linear. And this one have few associated proteins, while this one will have histones. Okay, and you can see here that if you go on and study in more detail, you will see that these associated protein in archaea are like histones. So even in this chromosome, you can start seeing more difference. So if you see it from very far, from very general, they seem to be very similar. But however, new tools have allowed us to study that in more detail. So I told you about the ribosomal RNA that they were different. When you go and study actually the cell walls, you see that the proteins, the lipids, the sugars that are involved in the cell walls are different. So while in bacteria you have a combination of proteins that are I cannot pronounce properly, peptoglycans, and here you have a variation of that, yeah, and also that one that is based on protein, so it's not the same. And in eukarya you will have several variations. Yeah, in plants you have cellulose and pectin. In fungi you also have a different one, and in animals you don't have one. Okay, and what I'm going to show you in the following slide is that those differences are much higher than you think, or that there are much more different. So although I show you all these green characteristics there, they seem to be very general, and they seem to be that bacteria and archaea are very similar. When you go and study in more detail each one of those, you will see that they are actually not as close as we thought, that they are just that they are similarities that occur in many cases because they are small. Yeah, so for example, if you have a small cell, you need a lot of DNA, so you don't have to pack it as well. Yeah, you have to pack it very, like using this linear chromosome, you can have just few protein associated. Okay, I'm not going to go into a lot of detail more. I already talked about that, but during the semester, you will see that in more detail. So now we are going to study in more detail some of this structure. And again, there is still a debate about that. But when you go and study the mechanism of processing of RNA, you see that bacteria has a minimal processing, archaea has a moderate, and eukaryotes has an extensive processing. If you study genetics, you know that there are so many proteins and molecules. If you study, for example, the transcription initiation, that it should be a one signal that it should be conserved during evolution. Yeah, that is very important. Bacteria have one, and eukaryotes have one. Yeah, so you see that some differences are emerging. When you study, for example, the polymerase, the RNA polymerase, again, one of the key difference of how cells start appearing, yeah, how this self-replication appear. Remember the RNA word. So you have ones that have bacterial type or ones that have some feature of bacteria and eukaryotic types, and this one will have a eukaryotic type. So again, if you see, there are just more variation when you study those in more detail. And again, if you see the RNA processing, the transcription initiation, this the copy, the proteins involved in copying the RNA. So those are very important mechanisms at the cellular level that they should be conserved. So the difference are more dramatic than we thought in the beginning. So the previous slide, as I said before, they were just features that they were very general. But when you go and study those structures in more detail, you see that there could be some difference. Here, you can also find some similarities. So, for example, the size of the ribosome and the number of proteins in RNAs. So you can see that they are similar between bacteria and archaea, and here you see that they are different in eukaryotes. The translation initiation, again, so you see that bacteria has one and eukaryotes will have another one. And the membrane phospholipids, again, there is a similarity here and similarity here and a difference here. So for that reason, what I wanted to tell you, I didn't want it to confuse you. I just wanted to show you that, again, the main idea of this is that you know the term prokaryotes. It doesn't mean that they are phylogenetically close related. When you go and study those the traits of those at the cellular level of these different domains, it seems to be more complicated than we previously expected. And based on the information that we have here, uh, it seems that these three domains, they are separated from each other. Yeah, and it's not that prokaryotes are close, bacteria and archaea are close to each other. Okay, hopefully, if you have any doubt, you can ask me some questions, or you can read the textbook that they have a lot of information. Now, we're going to have a question that is slightly more difficult, that is at the university level, and we're going to read it very carefully. So, what is a distinguishing feature, and now let's make the groups. Let me put it with a different color of archaea. So archaea is group number 1 compared to bacteria number 2 and eukaryotes. Okay, number 2. So how you can group bacteria and archaea, what they have in common that is different from archaea. Okay, so this is something that is relatively more difficult, and we're going to use the knowledge that we have from high school, from elementary school, and also some of the things that we learn. And let's see how far we can answer. So let's start with the first A, the presence of a true nucleus. So these two groups are not divided by the presence of a true nucleus. That is very obvious. Yeah, archaea doesn't have a nucleus and bacteria doesn't have a true nucleus. So this is wrong. Lack of cell walls. So we know that archaea has a cell wall and bacteria has a cell wall too. So this is not a way to differentiate these two. Yeah, these two groups. Now you have two different options, yeah, that are more difficult. One that is the unique membrane lipids that I have never talked about that, yeah, in the previous table. But if you read your textbook, you will see that one that is a feature that is very interesting to analyze, and another one that is use chlorophyll for photosynthesis. Okay, and definitely bacteria and eukaryotes perform photosynthesis. You have these cyanobacteria, and plants have photosynthesis. So that could be an option, yeah? Yeah, so it could be C or D. So if you read the textbook, you will see the answer. If not, I will give it to you in 1 second. So here the answer is that archaea has unique membrane lipids, and here you can see two differences. So here you have the diagram from the phospholipid of archaea, and here the phospholipid of bacteria and archaea. So if you see the second group, yeah, the bacteria and archaea had the membrane lipids are similar, and the archaea are different. So this is why we said that these three groups, they are the phylogeny. There are three domains that are very different. So let me show you when you compare here. This is the first difference. Well, uh, the just as a reminder, this is the carbon component, the long chain, and here is, well, you will see the phospholipids. Just as a reminder, I know you know that, but when you see the first thing is that here you have a side chain. So here there is a side chain, there is a side chain, there is a side chain, and here, if you see, there is no side chain. Okay, so there is one difference there. The second difference is that in the phospholipid area, you see here there is an ether linkage, if you remember your chemistry, while here in the bacteria and eukarya you have an ester linkage. Okay, so you can see it here very clearly. So I told you that archaea has a unique membrane lipids. Yeah, that the phospholipids are different from those found in bacteria and eukarya, and in two ways. In one way is through the side change, and in the other one was through the ether bonds. Okay, I hope that that was clear. Now, in the following slides, I'm going to show you that option D is incorrect. And the idea is that there will be difference in the pigment and the photosynthetic pigments between the three domains. Okay, so it's not a character that allow you to make this clear difference that you have one group here, A, and another group here, B. Let's suppose like that. So that bacteria is here with eukarya and archaea is here. What we have is that all of them, they have they could have different pigments used for photosynthesis. So, for example, we have generally talked that photosynthesis has chlorophyll, and it's true in plants you can find chlorophyll, and in cyanobacteria that is true. So that partially will say that is correct. Yeah. However, you in bacteria you can also find other pigments for chlorophyll. So when you have uh, purple bacteria, like purple bacteria, green sulfur bacteria, you can have something called bacteriochlorophyll. Okay, so in our words, they could be multiple pigments. So, and as a result, you can have different type of photosynthesis. One that is anoxy that don't require oxygen, while the other one that require oxygen. Okay, so again, this show that the correct answer will be the lipids, not the option D about the chlorophyll. And to finish this, here in Archaea, as I show you, this is unique. And here, Archaea again is also unique. Okay, so again, D is incorrect because the three domains can use different type of pigments for chlorophyll, although there could be some intersection between cyanobacteria and plants. So Archaea do not use chlorophyll for photosynthesis. Instead of that, they use a different pigment that is the bacteriorhodopsin. And what is cool about that is that, you know, if you study introductory, if you remember your introductory biology course, you will say, oh, this is pretty similar to what we study in photosynthesis. And yeah, there's generation of ATP, but in this case, this way of production of ATP don't require carbon fixation. Okay, so it's pretty interesting. So as a result, okay, so as a result, when we read this question and we said that what was the distinguished feature between the group number one, that it was Archaea, that let's put it here, yeah, group number one, and the group number two, that it was bacteria and eukaryotes, that it was the group number two, that it will be here, group number two, let me circle it. Yeah, the only trade that it was, the only option that it was possible, it was the unique membrane lipids, as I just explained. And I show you that although chlorophyll, yeah, can be used in both bacteria and eukarya, um, they can also bacteria that they are so diverse that some bacteria can use other type of pigment for chlorophyll. So this will be wrong. Okay, so hopefully that was more clear, and you can, if this question is in a quiz or in a test, you can answer it and it's not too complicated for you. So to finish this section and to summarize all the things that we said, so we know that bacteria, archaea, and eukaryotes, these three domains, they have difference in many different ways, and I show you that. And I show you that, okay, they eukarya and bacteria, they can share, for example, the lipids and the structure of the lipids in the membrane. Bacteria and archaea, they can share the size, yeah, and eukarya and archaea, they can have also uh, similarities in the initiation of the translation or transcription, yeah. So there are some similarities. However, each group is different from each other, yeah. So this is why we have one, two, three domains that definitely they diverge a lot from each other. Okay, I hope that you enjoyed that part. And with this, we are done with this part of this lecture.